Welcome. Today we're diving into the fascinating world of cell division, the process that allows living organisms to grow, heal, and reproduce. Whether you're curious about how your body repairs itself or how traits are passed down through generations, understanding cell division is key. We'll explore the structure of chromosomes, the phases of the cell cycle, and the two main types of cell division, mitosis and meiosis. By the end, you'll see how these microscopic events shape life as we know it. Let's begin our journey by understanding what cell division really means and why it's so essential for all living things. Cell division is the process where a mature cell splits to form new daughter cells. These daughter cells inherit many characteristics from their parent cell, ensuring continuity of life. This process is fundamental for growth, repair, and reproduction in all living organisms. Without cell division, life as we know it wouldn't exist. There are two main types of cell division: mitosis, which occurs in body cells, and meiosis, which is crucial for reproduction. Each type has its own unique role and significance, which we'll explore in detail as we move forward. Let's clarify the two main types of cell division. Mitosis is the process responsible for growth and repair in our bodies, producing two identical daughter cells from a single parent cell. On the other hand, meiosis is essential for sexual reproduction, resulting in daughter cells with half the number of chromosomes. This reduction is vital for maintaining the correct chromosome number across generations. Both processes are tightly regulated and play distinct roles in the life cycle of organisms. Understanding their differences is crucial for grasping how life perpetuates and evolves. Cell cycle is the series of events that cells go through as they grow and divide. It consists of two main phases: a long non-dividing phase called interphase and a short dividing phase known as the mitotic phase or M phase. Interphase is where the cell spends most of its life growing and preparing for division. The mitotic phase is when the actual division occurs. This cycle ensures that cells are ready and equipped to divide properly, maintaining the health and function of tissue. Interphase is often called the resting phase, but it's far from inactive. During this time, cells increase in size and volume, carrying out normal metabolic activities and preparing for the next round of division. Interphase is divided into three subphases: G1, S, and G2. Each of these plays a specific role in ensuring the cell is ready for the critical process of division. Let's take a closer look at what happens in each subphase. First subphase of interphase is the G1 phase, or first growth phase. Here, the cell grows rapidly and performs its normal functions, such as metabolism and protein synthesis. This phase is crucial because it sets the stage for DNA replication. If the cell doesn't grow properly or encounters problems, it may not proceed to the next phase. The G1 phase ensures that only healthy, well-prepared cells move forward in the cycle. Next comes the S phase, or synthetic phase. During this stage, the cell duplicates its DNA, ensuring that each daughter cell will receive a complete set of genetic material. DNA replication is a highly regulated and precise process, as any errors can lead to mutations or cell malfunction. The successful completion of the S phase is essential for the integrity of the genetic information passed on to new cells. Final subphase of interphase is the G2 phase, or second growth phase. During G2, the cell continues to grow and makes final preparations for mitosis. It checks for any DNA damage and repairs it if necessary, ensuring that the cell is fully ready to divide. This phase acts as a quality control checkpoint, preventing damaged or incomplete cells from proceeding to division. Now let's move on to mitosis, the process by which somatic or body cells divide. Mitosis results in two identical daughter cells, each with the same number of chromosomes as the parent cell. This process is also called equational division because the genetic material is equally distributed. Mitosis is essential for growth, repair, and replacement of cells in multicellular organisms. It consists of two main processes: karyokinesis, the division of the nucleus, and cytokinesis, the division of the cytoplasm. Karyokinesis is the first major step in mitosis, involving the division of the cell's nucleus. This process ensures that each daughter cell receives an identical set of chromosomes. Karyokinesis itself is divided into four distinct phases: prophase, metaphase, anaphase, and telophase. Each phase plays a unique role in the accurate separation of genetic material. Let's break down what happens in each phase, starting with prophase. During prophase, the chromatin fibers in the nucleus condense and become visible as thick chromosomes. The nucleolus and nuclear membrane disappear, signaling the start of division. Meanwhile, centrioles duplicate and the spindle apparatus begins to form, setting the stage for the chromosomes to be moved and separated. This phase is all about preparing the cell for the precise distribution of genetic material. Metaphase, chromosomes are arranged along the metaphase plate, also known as the equatorial plane of the cell. This alignment ensures that, when the chromosomes are pulled apart, each new cell will receive an identical set. The spindle fibers attach to the centromeres, holding the chromosomes in place and preparing for the next critical step: separation. Anaphase is the phase where the real action happens. The centromeres divide and the sister chromatids are pulled apart by the spindle fibers toward opposite poles of the cell. This separation ensures that each daughter cell will receive an exact copy of the genetic material. The movement is quick and precise, highlighting the cell's remarkable ability to organize and distribute its contents. During telophase, the spindle apparatus disappears and the chromosomes begin to uncoil, returning to their thread-like chromatin form. Nuclear membranes and nucleoli reappear around the two sets of chromosomes, marking the end of nuclear division. The cell is now almost ready to split into two, with each new nucleus containing an identical set of genetic material. After the nucleus divides, the cell undergoes cytokinesis, where the cytoplasm is split to form two new daughter cells. In animal cells, this happens through the formation of a cleavage furrow that deepens until the cell is pinched in two. In plant cells, a cell plate forms at the center, eventually developing into a new cell wall. Both methods ensure that each daughter cell receives the necessary cellular components to function independently. Mitosis is more than just a way to make new cells. It's essential for the growth of organisms, allowing them to increase in size and complexity. It also plays a crucial role in repairing damaged tissues and replacing old or worn-out cells. Without mitosis, our bodies couldn't heal wounds or maintain healthy tissues. This process keeps us growing, healing, and functioning every day. Now let's shift our focus to meiosis, the type of cell division responsible for producing gametes, sperm, and egg cells. Unlike mitosis, meiosis reduces the chromosome number by half, creating haploid cells from a diploid parent. This reduction is vital for sexual reproduction, ensuring that offspring have the correct number of chromosomes. Meiosis introduces genetic variation, which is the foundation of evolution and diversity in living organisms. Meiosis is unique because it involves two successive divisions, meiosis I and meiosis II. The first division, meiosis I, is called reductional division because it halves the chromosome number. The second, meiosis II, is similar to mitosis and separates the sister chromatids. Together, these divisions produce 4 haploid cells from one diploid parent cell, each with a unique combination of genetic material. Meiosis I is the stage where the chromosome number is halved. It consists of 4 phases: prophase I, metaphase I, anaphase I, and telophase I. Each phase plays a specific role in ensuring that the resulting cells have half the original chromosome number. This division is crucial for maintaining genetic stability across generations and for introducing genetic diversity through processes like crossing over. Prophase I is the longest and most complex phase of meiosis. It's divided into 5 substages: leptotene, zygotene, pacotene, diplotin, and diakinesis. During these stages, chromosomes condense, pair up, and exchange genetic material through crossing over. This exchange creates new genetic combinations, which are vital for evolution and diversity. The nuclear membrane and nucleolus disappear and spindle fibers begin to form, preparing the cell for the next step. In the leptotene stage, the chromatin network condenses and chromosomes become visible. Centrioles duplicate, setting the stage for the pairing of chromosomes in the next substage. This condensation is the first visible sign that the cell is entering meiosis and preparing for the complex process of genetic recombination. During zygotene, homologous chromosomes begin to pair up, a process called synapsis. This pairing is essential for the exchange of genetic material that occurs later. The chromosomes align closely, setting the stage for the crossing over that will increase genetic diversity in the offspring. In the pacotene stage, chromosomes remain paired at points called chiasmata where crossing over occurs. This is when segments of genetic material are exchanged between homologous chromosomes, creating new combinations of genes. Crossing over is a key source of genetic variation, which is essential for evolution and adaptation. During diplotin, homologous chromosomes begin to repel each other, except at the chiasmata where they remain connected. This separation is important for the eventual distribution of chromosomes into daughter cells. The chromosomes are now ready for the final preparations before division. In diakinesis, the nuclear membrane and nucleolus disappear and spindle fiber formation begins. The chromosomes are fully condensed and ready to be moved to the center of the cell. This marks the end of prophase I and the beginning of the next phase, metaphase I. During metaphase I, homologous chromosomes are arranged at the equatorial plane of the cell. This alignment ensures that when the chromosomes are separated, each new cell will receive one chromosome from each pair. The spindle fibers attach to the chromosomes, holding them in place for the next step. In anaphase I, homologous chromosomes, after crossing over, start moving to opposite poles of the cell. This separation reduces the chromosome number by half, ensuring that each daughter cell will be haploid. The movement is guided by the spindle fibers, which pull the chromosomes apart with precision. Telophase I marks the end of the first meiotic division. The nuclear membrane and nucleoli reappear around the separated chromosomes, completing the division. Each resulting cell now has half the original chromosome number, setting the stage for the second meiotic division. Meiosis II is similar to mitosis in its process. It separates the sister chromatids in each of the 2 haploid cells produced in meiosis I. The result is 4 haploid daughter cells, each with a unique combination of genetic material. This division is crucial for generating genetic diversity in sexually reproducing organisms. Meiosis is vital for sexual reproduction. It restores the original number of chromosomes in offspring, introduces genetic variation, and provides the foundation for evolutionary processes. Without meiosis, species would not be able to adapt and evolve over time. The genetic diversity created by meiosis is what allows populations to survive changing environments and challenges. Let's compare mitosis and meiosis side by side. Mitosis occurs in somatic or body cells and produces 2 identical diploid daughter cells. Meiosis, on the other hand, occurs only in reproductive cells and results in 4 haploid daughter cells. Mitosis involves 1 division, while meiosis involves 2 successive divisions. The genetic material in mitosis remains the same, but in meiosis, it's halved and shuffled, increasing diversity. Summarize: mitosis and meiosis differ in several key aspects. Mitosis occurs in somatic cells, involves 1 division, and produces 2 diploid cells identical to the parent. Meiosis occurs in reproductive cells, involves 2 divisions, and produces 4 haploid cells with half the chromosome number. These differences are crucial for understanding how organisms grow, repair, and reproduce. Chromosomes are the carriers of genetic information. They play a vital role in heredity, variation, and the evolutionary development of species. Discovered by Walter Fleming in 1882, each chromosome contains a centromere that holds together two sister chromatids. The position of the centromere determines the chromosome's shape, and chromosomes can be classified into four morphological types based on this. Humans, chromosomes are classified into two types based on their function: autosomes and heterosomes. Autosomes are non-sex chromosomes responsible for most of the body's traits. Heterosomes, also known as allosomes or sex chromosomes, determine the sex of an individual. This distinction is crucial for understanding inheritance patterns and genetic disorders. Chromatin is the material that makes up chromosomes. It consists of DNA and proteins called histones. The DNA strand winds around a core of 8 histone molecules, forming a structure known as a nucleosome. This packaging allows long DNA molecules to fit inside the cell nucleus and plays a role in gene regulation. DNA, or deoxyribonucleic acid, is a large molecule made up of two strands twisted around each other in a double helix. These strands are complementary, meaning the information on one matches the other. DNA stores the genetic instructions for building and maintaining an organism, making it the blueprint of life. The basic unit of DNA is the nucleotide. Each nucleotide consists of 3 parts: a phosphate group, a deoxyribose sugar, and a nitrogenous base. There are 4 types of nitrogenous bases in DNA: adenine A, cytosine C, thymine T, and guanine G. The sequence of these bases encodes genetic information. DNA, the nitrogenous bases pair in a specific way. Adenine pairs with thymine and cytosine pairs with guanine. This base pairing is essential for the accurate replication and transmission of genetic information. The complementary nature of the strands ensures that, when DNA is copied, the new molecules are identical to the original. DNA replication is the process by which a cell makes an exact copy of its DNA. This is crucial before cell division, ensuring that each daughter cell receives a complete set of genetic instructions. The process involves unwinding the double helix, separating the strands, and building new complementary strands using the original as a template. DNA replication involves several key steps. First, the double helix unwinds and the strands separate. Then new nucleotides are added to each original strand following the base pairing rules. Enzymes help assemble the new strands, resulting in 2 identical DNA molecules. This process is highly accurate, but occasional errors can lead to mutations. Genetic variation is the raw material for evolution. Processes like crossing over during meiosis and occasional mutations during DNA replication introduce new genetic combinations. These variations can be beneficial, neutral, or harmful, but they provide the diversity needed for populations to adapt and evolve over time. Proper cell division is essential for health. Errors in mitosis or meiosis can lead to genetic disorders, cancer, or infertility. The body has many mechanisms to ensure accuracy, but when these fail, the consequences can be serious. Understanding cell division helps us appreciate the complexity of life and the importance of cellular health. Knowledge of cell division is crucial in medicine. It helps doctors understand diseases like cancer, where cell division goes out of control, and guides treatments such as chemotherapy. It's also important in genetic counseling, fertility treatments, and regenerative medicine, where understanding how cells divide and differentiate can lead to new therapies. Cell division isn't just a topic for scientists, it affects us all every day. From healing a cut to growing taller or even fighting off infections, cell division is at work behind the scenes. Understanding these processes helps us appreciate the complexity and resilience of living organisms, including ourselves. Summary: cell division is a fundamental process that drives growth, repair, reproduction, and evolution. From the structure of chromosomes and DNA to the intricate steps of mitosis and meiosis, every detail is finely tuned to ensure life continues. By understanding these processes, we gain insight into the very foundation of biology and the remarkable complexity of living things. Thanks for joining this exploration into the world of cell.